On this beautiful day, I am pondering enzymes and considering both the root of the word and also the beauty of the efficiency of these intricate machines. So enzymes roots from the word in yeast. And in fact, quite literally, when people started to think about enzymes, they were arguing vehemently that the reactions that were made possible by yeast were either reactions that required the entire cell to be present in order for them to occur, or that could occur if only the enzymes from the cell or some component, right, from within the cell were taken out, that those reactions could be made possible. So these were two schools of thought. In fact, they were called the vitalists, who said that the cells were needed in order to make the fermentation possible, and the mechanists, who said, oh, you can take out some component from within the cell, turning out to be enzymes, and that will allow these reactions to be catalyzed. They actually fiercely fought with one another, and they published cartoons that would show yeast taking in sugar in one orifice and then basically pooping out ethanol, like producing alcohol from the other orifice. And the truth be told, both the vitalists and the mechanists, to a certain extent, were correct. Because yes, it was the enzymes from within the cell that enabled those reactions to take place. But we could also argue that within a cellular system, right, like the vitalists, we could see that those reactions maybe are nurtured the most effectively. Lots of discussion. Maybe I'll take you out for coffee and we'll talk more about that. But the term enzyme just does root from this in yeast definition or origin. So enzymes are catalysts. They do not change the equilibrium of a reaction. So if a reaction is not favorable, an enzyme doesn't make it favorable. But enzymes do act to speed the rate of the reaction. And they do that in a couple of ways. But ultimately, they stabilize this very precarious, very awkward transition state and enable it to decrease its overall awkwardness, right, and decrease the energy of that awkward transition state. What's really cool about enzymes is that, unlike chemical catalysis, in which we tend to have to think about maybe bringing temperatures to a very high temperatures that we wouldn't want to live at, or we change the acidity of the environment. And I'm kind of jokingly showing a picture here of Norris Geyser Basin that's both acidic and extremely hot. We think about that as being the conditions that you need for chemical catalysis. But enzymatic catalysis is more like a day at the spa. I mean, these are nice, nurturing, very mild conditions that are required for enzymatic catalysis. Said another way, the kind of conditions that we maintain within our body: physiological pH, physiological temperature. So enzymes do what they do in very mild conditions. And enzymes are pretty specific. That is to say that enzymes typically have one specific substrate that is most attractive to them. So next time you're in the bar, you can try the pickup line: if I were an enzyme, I'd want you to be my one specific substrate. And some enzymes are genuinely that way. That is, they really only recognize a specific substrate. For example, if they act on L-glutamate, they're not going to act on D-glutamate. So the stereoisomer is out, right? And that's true of many enzymes, that they exhibit stereospecificity. But enzymes differ. Just like people, right? Some are more promiscuous than others. Some have more things that they recognize as their specific substrate. A good example, an awesome example, is the enzyme papain. Papain breaks peptide bonds in polypeptide chains, and it is nonspecific about that. It doesn't care if it's breaking a bond between glycine and alanine or a bond between arginine and aspartate. It will break those bonds equally specifically, if you will. It's more promiscuous. But there's an enzyme, another digestive enzyme that we'll learn about soon, called trypsin, and it's more specific about which peptide bonds it will break. So enzymes have high reaction specificity and they lack formation of wasteful byproducts. I had a good friend who used to always tell me, Rachel, you're a byproduct of the 80s. And I would look at him and say, like, wait a minute, don't I at least warrant a product of the 80s? So when we think about non-enzymatic catalysis, often our product yield is not particularly high. But man, with enzymes, essentially 100% purity of products. So we really do see enzymes being the most efficient machines, which is why this field of biomimicry is so very important. Because if we could do if we could do energy generation the way that enzymes do generation of energy, we would be in business. I mean, enzymes have nearly 100% purity of products. Enzymes also can couple reactions. This is wicked cool. That is to say, a high energy yielding reaction like the hydrolysis of ATP can be coupled to an energy consuming process like the generation of glucose 6-phosphate. So here you can see that coupling. ATP is hydrolyzed, powering the phosphorylation of glucose, an energy generating reaction coupled to an energy consuming reaction. And that's a pretty cool thing. This enzyme that does this, I grabbed this off of Chimera, took it from Protein Data Bank to NZT. This is hexokinase. Hexokinase meaning a phosphorylating enzyme, kinase, that recognizes 6 carbon sugars. We'll meet it later in all its full glory. When enzymes were first being elucidated and understood as binding to a specific substrate, Emil Fischer proposed this idea of an enzyme binding its substrate as being like a key fitting into a lock, where the lock is the enzyme, the key is the substrate. This is an okay way to think about enzymes, and it worked for modeling for a while, but in fact, the actual way that enzymes tend to work tends to be more like, you know, when you get a brand new pair of like sleek driving gloves and you slip your hand into the glove and it fits around your hand. Like, it didn't quite look like your hand before you put your hand in. This is like the idea of induced fit. So induced fit is really how enzymes tend to fit their substrate. Hexokinase is a great example. When glucose binds to hexokinase and ATP, it wraps around them like a sleek driving glove would wrap around the hand. So enzymes also increase reaction rates immensely. Most reactions in a cell wouldn't occur to a much appreciable extent without an enzyme. Say, for example, the mere hydration of CO2. So the mere hydration of CO2 by carbonic anhydrase has around a 10 to the 7th increase in that reaction rate, but it wouldn't occur to much appreciable extent without the enzyme. So enzymes can increase reaction rates by 10 to the 3rd to 10 to the 20th. Again, as we said, it doesn't make an unfavorable reaction suddenly favorable. It just lowers that activation energy. Enzymes are often named by adding ase. So, you know, if you have something that is an enzyme involved in a redox reaction, it's dehydrogenase, protease, right? We can see that lipase, those that cleave lipids, those that cleave protein, peptidase, those that cleave peptide bonds. You just add ase on there. Enzymes often serve as points of regulation in metabolic pathways. We're going to get down and dirty with that idea that we might have an initial energy source that's converted to an intermediate, and then that intermediate is converted to another intermediate, and that intermediate to another intermediate, and enzymes are sort of the damming points along the way in that metabolic pathway. So they're points of regulation. An enzyme that we're going to get to know and love this semester is alcohol dehydrogenase. Alcohol dehydrogenase is our hero because it is the enzyme in our liver that detoxifies ethanol and allows us to break down that toxin, if you will. So if you're praying to the porcelain goddess, you're actually praying to alcohol dehydrogenase.